So, just an additional question: What do you, what's the experience with mutations of the coronavirus and the monoclonal antibodies? There's a little bit of data, I think, out on that.
So, all these variants of concern have been problematic for some of the monoclonal antibodies, particularly bamlanivimab and etesevimab, because there were certain mutations, such as the beta mutation and gamma mutation, which actually were resistant to both the bamlanivimab and etesevimab. And so for a while, we actually didn't give those at all. It wasn't allowed to give those that cocktail, and we only had the casirivimab and endevimab. And so as the Delta variant came up, the bamlanivimab and etesevimab are functional with the Delta variant. So a lot of states where the variants that are resistant are less than 5% of the overall variants out there, we can now use the bamlanivimab and etesevimab in those states now. And so the public health is monitoring these variants that are coming along and really adjusting the strategy of the monoclonal antibodies depending on what variants are out there that are resistant to the monoclonal antibodies. And a lot of these monoclonal antibodies, for example, the casirivimab and endevimab, as well as the bamlanivimab and etesevimab, they're cocktails because they hit two different epitopes on the spike protein. And the idea there is that you have to have multiple mutations in order to knock both of those antibodies off the spike protein and prevent them from binding. And it's really just random chance, and that did happen again with certain variants for the bamlanivimab and etesevimab. But by having two of the monoclonal antibodies, it reduces that chance of having resistance. Now, when you look at sotrovimab, that's a single monoclonal antibody. And how that was developed was that they actually identified that to have activity against SARS-CoV-1. And so it actually cross-reacts against both SARS-CoV-1 and SARS-CoV-2. And so the idea is that if you have an epitope that is conserved across these different viruses, that it likely is an epitope that is less likely to mutate because it has some kind of function that can't be mutated. And therefore, with this one single monoclonal antibody, the fear of building resistance to that is less.